Okay, if you are doing honors bio as a freshman, there's a couple extra things you're going to need to know if you're not currently in advanced science. So I'm going to detail a couple pieces of information for you, so you guys will be familiar with these when you hit the honors class next year. So to do a quick review, you have seen a picture of a nucleotide, and by now you have built your DNA model. So you have the phosphate, the 5-carbon sugar, and the nitrogenous base. Okay, the nitrogenous base could be a G, an A, a T, or a C. And so we use whichever base you're dealing with to name the entire nucleotide just to make our lives easier. So there's a couple specifics about our bases, first of all, and I'm going to go into those next. So your base could be a G, an A, a T, or a C, right? So you guys are familiar by now that the G's, or the guanines, can only bond with the cytosines, or the C's. And the adenine, or the A's, can only go with the thymines, or the T's. And why is that? So first of all, you're going to see another term here underneath the diagrams. And so if we go to the very bottom, A and G are known as purines. And a purine is just the chemical structure where we have a pentagon joined up with a hexagon here. And so you'll see that both of these are purines, which means they have those pentagons and hexagons. Whereas we have pyrimidines up here. Pyrimidines are just hexagon chemical structures that have some subtle differences in their atoms, but we'll get to that in a sec. So the gist is that a purine does not pair up with another purine. The purine chemically can only bond with another pyrimidine. Now, if that's the case, well, then why can't guanine switch teams and bond with thymine? That's a purine to pyrimidine, so why can't that work? Well, there's another couple differences. If we look at our pyrimidines first, so thymine and cytosine, let's look at the shapes here and look at any differences we have. That's the same. That's the same. The nitrogen right there is the same. However, we have an H bond there. But the differences come in these two spots. And so a thymine has a CH3 group, which is called a methyl group. But the big difference is that it has an O there. Now, instead of an O, or an oxygen on top of the pyrimidine structure, if we go to the cytosine, it instead has an amine group, or an NH2. And so the fact that you have a purine to pyrimidine bond going on is also a characteristic that you need at least one O and one NH2 in your bonds. So what do I mean by that? Well, if we look at the guanine, who is cytosine's partner, guanine on the top of the purine structure has an O there. The O is what needs to bond with our NH2. So every base pair needs an NH2 and an O to make things work. But if we look at the other purine, which is adenine, it doesn't have an O there. Instead, on top it has an NH2. And, well, who's on top of the thymine in DNA? It's not an NH2 like our cytosine was. It's instead an O. So we have the NH2 here and the O there, which means that these guys can now bond. So it's not only just you need to have a purine bonding with a pyrimidine, but it's also that they need to have at least one O group and one NH2 group. And that's why A can only go with T, or C can only go with G. It's because of those chemical characteristics, which are very important. So the gist is purines can only bond with pyrimidines, so remember that. Now another thing is about the carbon in DNA, or the sugar in DNA. It's a pentagon, and we have 5 carbons here. So why is DNA called DNA? Well, it stands for deoxyribonucleic acid. And if we look at our ribose sugar, we have a little bold highlighted OH there. Deoxyribose means that the ribose has had the oxygen taken off. It's been deoxygen. Whoops. Oh dear. It's been deoxygenated. So we get rid of that. And instead, we don't have that oxygen over here. We just have an H. So that's why it's called deoxyribonucleic acid, because we took the oxygen off that sugar. Now here's the big deal. It's a 5-carbon sugar, carbon 1 here, 2 here, 3 here, 4 here, and 5's hanging off the top left side because this is an oxygen. So the most important carbons in deoxyribose are carbon 3 and carbon 5. So let's talk about why. If we go down to a picture of a nucleotide and we find our sugar here, we have deoxyribose there and there. Now our carbons. This is carbon 1, 2, 3, 4, and 5. 1, 2, 3, 4, and 5. 3 and 5 are the most important because they connect the chain. So if I highlight our phosphate group in a little circle and make things a little easier, we notice that the 5 carbon connects to the phosphate up top, and the 3 carbon connects to the phosphate on bottom. And then the connection perpetuates, so it continues. We have a connection at the 5 and the 3, and then it keeps going. We'd have another phosphate group there, and we are able to build the chain. Now, the other thing is that which carbon is on top of this sugar? I'm looking at it right side up, so who's on top, the 3rd or the 5th? The 5th is on top of the 3rd, and it's going to be like that on every sugar. 5 is on top of 3. And so what we call this is we call this the 5 to 3 direction side of DNA, because the 5th carbon of every sugar is sitting over or on top of the 3rd carbon of the sugar. So the 3 and carbon of the sugar is always going to be on bottom. So we call this the 5 to 3 direction. Now in this case, when we built our models, we had to have it flipped over. The reason you have to have the other side flipped over is so the nitrogen base pairs can bond, otherwise they won't be able to bond. And to make that happen, our sugar gets flipped over, so the oxygen on top is now facing the bottom side. So let's label our carbons. 1, 2, 3, 4, 5, and so on. 1, 2, 3, 4, 5. Now in this case, the sugar's flipped over, so who's on top here? The 3rd carbon or the 5th carbon? The 3rd's on top. And it's always on top of every sugar because this whole side's flipped over. So in this case, we're going to call this side of DNA the 3 direction to 5 direction. So it's going the other way. And so the whole theme about how DNA is flipped on one side is actually called anti-parallel. And when you do, what does anti-parallel mean exactly? Anti-parallel, you see it all the time. It's when you're driving on a road, and this is you in a car, and this is somebody else in a car. You guys have parallel paths, but you're going opposite directions. You're going that way, they're going that way, but you're still parallel to each other. And so that's called anti-parallel, which describes the fact that one side of DNA is flipped over. And so when you look at a picture of DNA and you see this 3' and 5', all that means is that on this backbone of the DNA, the 3rd carbon of every sugar is going to be on top. And so we're looking at the sugar flipped upside down on that side, whereas the 5th carbon is always going to be on bottom. And then if we look at the other side, it's labeled 5' because this sugar's right side up. So the 5th carbon is on top of the 3rd every time. So that sugar's right side up and it's not flipped upside down. And so that shows us that DNA has its anti-parallel structure. So the gist is you're going to see a 5 and 3 in Honors Bio. So I want you to understand that all it means is that we're dealing with the 5th and the 3rd carbons because they form the chain of phosphate to sugar to phosphate to sugar. And to be able to make DNA parallel, the sugar's flipped over on one side. And so one side will go 5 to 3, and then the flipped over side is going to be 3 to 5 because the sugars are upside down. That is all.